In today's video, we'll be studying the cell under the heading of the plasma membrane and the cytoplasm. So here we have the outline of all we'll be discussing: the definition of the cell, cell theory, plasma membrane, cytoplasm, under which we'll consider the cytosol, cellular inclusions, as well as the organelles of the cell. So first of all, let's start with the definition of a cell. We're very familiar with the definition of a cell, which is that the cell is the basic structural and functional unit of life. This simply means that the cell is the smallest unit that can carry out the general life functions. So we're probably familiar with the general life functions of movement, respiration, nutrition, irritability, excretion, reproduction, and the rest of the part where we now consider competition, habitat, adaptation, and of course death. So a cell is the smallest unit that can carry out all of this function, okay, and it's also the smallest unit that has that structure of life. So that's basically what the definition of the cell means: the basic structural and functional unit of life. When we begin our study on cells, we would learn that cells have all of these organelles and they have all of these structures, they have the cell membrane and so on. But what we're studying is actually a generalized cell. Now, what exactly is a generalized cell? A generalized cell has all of these organelles: the mitochondria, the smooth endoplasmic reticulum, rough endoplasmic reticulum, and so on. However, as we would go on to see, not all cells have all of these organelles. Okay, some cells have more of a particular organelle than another. Say for instance, some cells are very metabolically active, and this would mean that they would have more mitochondria. Okay, because as we would go on to see, the function of the mitochondria has to do with energy, okay, the release of energy and so on. Alright, now let's take a look at the cell theory. There are three basic concepts under the cell theory. First of all, just remember the definition of a cell, okay? So the definition of a cell forms one of the concepts of the cell theory. The next is, where are cells found? Okay, so where can they be found? Where? So the first concept would be the definition of the cell. The next concept would be, where can the cells be found, or where can cells be found? And the third concept of the cell theory is, how are they formed? We had already defined the cell as the basic structural and functional unit of life. So the first concept of the cell theory is that cell, or rather a cell, is the basic structural and functional unit of life. Now, I said the next concept, or the second concept of the cell theory, has to do with, where can cells be found? So where can cells be found? All living things are made up of cells, okay? All living organisms are made up of cells. This is the next concept of the cell theory. And the last concept of the cell theory, how are cells formed, is that all cells originate from pre-existing cells. So these are the three concepts of the cell theory. In this video, of course, we'll be considering a generalized cell. And a generalized cell has three major parts. Okay, so there are three major parts of a generalized cell. A generalized cell has the plasma membrane. This is sometimes called the cell membrane or the plasma lemma. It also has the cytoplasm, and lastly it has the cell nucleus. Okay, these are the three major parts of a generalized cell. However, for this video, we'll be considering only the plasma membrane and the cytoplasm. In a subsequent video, we'll discuss the cell nucleus in details. Okay, now let's take a look at the plasma membrane. Now, the plasma membrane is what forms the border around a cell. We have the extracellular fluid, and then we have the border formed by plasma membrane, and then we have the intracellular fluid. The plasma membrane, of course, is sometimes called the plasma lemma, or in some cases it's called the cell membrane. Now, just before we continue on our study of the plasma membrane, it's very good for us to take note of this very important fact. Okay, now generally when we're considering a living organism, and we would say that this living organism has a level of organization, okay, and the level of organization to this organism or for this organism would be that, well, we have the living organism, and this living organism is made up of a bunch of systems. Okay, so we have biological systems, and these systems are made of organs that work together. The organs are made of tissues, and then we have the tissues being made up of cells. Okay, so we can see that the smallest unit of the living organism would be the cell. Now we can make a general conclusion that a living organism is simply a bunch of cells. However, this would be an overgeneralization, because although the living organism is indeed a bunch of cells, there are other substances that are not necessarily cellular that make up the general mass of the living organism. These substances that are not necessarily cellular but form part of the mass of the organism are referred to as extracellular materials. There are about 3 elements of the extracellular materials. Firstly, we have the extracellular fluid. We also have, forming part of the extracellular material, we have cellular secretions, and lastly we have the extracellular matrix. Now, the extracellular fluid is made up of a lot of dissolved substances that bathe the cell. It kind of serves as a nutritive juice to the cell, just surrounding the cell. And of course, we have cellular secretions. These are secretions that are released from the cell. As we would go on in our study, we would get to study epithelial tissues. Okay, so of course cells come together to form tissues. When we study epithelial tissues, we'd get to see that we also have epithelial glands. The secretion of these glands are from the epithelial cells. So we have these cellular secretions. This cellular secretion could be hormones and so on. Lastly, for the extracellular matrix, it's a jelly-like substance. So we'd get to see more of the extracellular matrix when we study connective tissue. In as much as a living organism is indeed made up of cells, there are also some materials that are outside the cell, and these are some examples. They fall under extracellular fluids, where we have these dissolved ions, electrolytes, proteins, and so on. We have cellular secretions as well as the extracellular matrix. So let's go back to our study of the plasma membrane. Okay, so as stated earlier, we have the plasma membrane serving as a barrier between the extracellular fluid and the intracellular fluid. The plasma membrane or the cell membrane is made of phospholipids, but not entirely phospholipids. There are 3 major macromolecules that make up the plasma membrane. We have proteins making up part of the plasma membrane, we have carbohydrates, and of course we have lipids. All of these macromolecules serve different purposes in the plasma membrane. Let's take a look at the function of each of the macromolecules. Firstly, would be considering the membrane lipids. The lipids form a wide class of macromolecules. However, the major lipids that form part of the plasma membrane are the phospholipids, and then we also have cholesterol. So these are the 2 membrane lipids that form part of the plasma membrane. So firstly, we have the phospholipid bilayer of the cell membrane. The phospholipid has a hydrophilic head and it has a hydrophobic tail. It's represented something like this. So we'd have the tail, and of course this would be the head. The head of the phospholipid is hydrophilic, meaning it's water loving. However, the tail is hydrophobic. So this would be the head, and then this would be the tail. The phospholipids orient themselves in such a way that the head faces the extracellular fluid on one side, and of course it's a bilayer, so it forms kind of like a sandwich of phospholipid. We have the head facing the extracellular fluid on one side, and the other side of the head faces the intracellular fluid. So we stated earlier that the head of the phospholipid is hydrophilic. Okay, so it's water loving. Generally, the extracellular and intracellular fluid are made up of water, and this hydrophilic head is able to interact properly with the water. So there's a hydrophilic interaction between the head of the phospholipid and the water-based extracellular fluid and intracellular fluid. And of course, the tail being hydrophobic is able to orient itself inwards. Okay, so it's able to keep away from the water environment. Okay, so it's very important for us to take note of this. Being a lipid, it's impermeable to water soluble substances. So substances that are generally water soluble would be unable to dissolve in the lipid and be unable to get through. However, some small ions can actually find their way through to the other side. As we would go on to see in subsequent videos, we could have gated channels, we could have pumps, membrane pumps, and we could have carriers. All of these are actually proteins, the gated channels, aquaporins, and all of those things. Membrane transport would not be considering that in this video. We also have things like exocytosis and endocytosis, and how some substances are able to basically leave the cell through the plasma membrane. So very important for us to take note of is the fact that the phospholipid bilayer acts as a semi-permeable barrier, and of course it delimits the cell. Okay, so it determines the area, so to say, that the cell occupies. So certain substances are able to pass through this phospholipid bilayer, and other substances are prevented from passing through, and hence it's a semi-permeable membrane. Alright, now taking a look at the other lipid that we have, which is the cholesterol. Generally, the phospholipid bilayer is kind of fluid. As we're going to see, the idea of the fluidity of the cell membrane or the plasma membrane is a theory known as the fluid mosaic theory. The plasma membrane, which is made of this phospholipid, is assumed to have kind of like a fluidity to it. Cholesterol helps to kind of like stiffen the plasma membrane. It's kind of like when you have a runny substance. Okay, maybe you have a runny soup and then you add cornstarch to it to kind of like thicken it, to change the consistency. That's kind of like what cholesterol does to the plasma membrane or to the cell membrane. Now back of course to the theory, the fluid mosaic theory. The fluid mosaic theory is the idea that the plasma membrane is fluid, and the proteins, as we'd go on to see, would study the membrane proteins, and these membrane proteins are able to swim through. It's actually called lateral drift. Okay, so the membrane proteins are able to drift, or they are able to move through the fluid plasma membrane. Now let's take a look at the membrane proteins. Generally, there are two types of membrane proteins. We have the integral membrane proteins and the peripheral membrane proteins. Now, integral membrane proteins are integral. They are very important to the cell membrane, and usually they exist as transmembrane proteins, but not in all cases. So a transmembrane protein would transverse through the plasma membrane. Okay, so we have phospholipid, and then we could have a plasma membrane protein transversing through the entire width of the plasma membrane. This could be an example of a transmembrane protein. However, in other cases, it could just be embedded on one side. Okay, something like this. Taking a look at this structure, we can see how integral the integral proteins are. Literally, they are integral to the structure of the plasma membrane, and this simply means that if you would have to remove the integral proteins, is as good as just destroying the entire plasma membrane, and by implication destroying the entire cell. Because taking a look at this portion, for example, this portion of the plasma membrane, you would notice that there's no phospholipid occupying the position of the transmembrane protein, for example. So if we were to take out this transmembrane protein, notice that there would be no phospholipid to occupy its position. So it's as good as destroying the entire plasma membrane and the cell by implication. For peripheral membrane proteins, just as the name implies, they are on one side of the plasma membrane, kind of like at the edge. So they could be either on the side of the extracellular fluid or the intracellular fluid, and they are peripheral proteins. And in addition to being peripheral, removing the peripheral protein would not exactly tamper with the overall structure of the plasma membrane. Okay, so generally proteins serve as transporters. They could serve functions of signal transduction. A hormone, for example, could insert itself at the receptor site formed by this protein, and then the signal would be transferred into the cell for certain actions to be taken. Lastly, under the plasma membrane would be for us to consider the membrane carbohydrates. Particular carbohydrates that actually form part of the membrane would be the oligosaccharide. The oligosaccharide really attach to proteins. So when they attach to proteins, they form glycoproteins, and when they attach to lipids, okay, so they could attach to the phospholipid. In that case, of course, it's a phospholipid, so it has a phosphate group attached to it. So this is the representation of the phospholipid that we have been considering so far. Now, when we have a glycolipid, an oligosaccharide chain replaces the phosphate group. This would be how carbohydrates come to play as membrane carbohydrates, and usually they serve for cell-to-cell recognition. Now, different cells have different pattern of arrangement of the oligosaccharide chain. So all of this oligosaccharide chain, of course, they are on the extracellular surface. Okay, so we could have the chains like this. Now, all of this oligosaccharide chain form what we call the cell coat or the glycocalyx. They serve cell-to-cell recognition, and this is very important because the pattern of the oligosaccharide chain differs for different cells. Now, the immune cells or the immune system is able to use the pattern of this cell code to determine when a cell is normal and when a cell is pathogenic. Okay, so when the immune system identifies the oligosaccharide or the glycocalyx pattern of a particular cell, it's able to tell whether it's a foreign cell. Okay, so of course, when it's a foreign cell, the immune system is able to destroy the cell. Just as a side note for us to take note of is cancer cells. Now, when a cell is starting to become cancerous, it's very interesting. Of course, we're not dealing with cancer cells in this video or cancer, but very interesting is the fact that the cancer cell almost continually changes its glycocalyx pattern. Take for instance, we have the immune cells. So we have the immune cells, and the immune cells have identified the glycocalyx pattern of this cell. So they have identified the glycocalyx pattern of this cell, and they have identified it to be a foreign cell, and so they make an attempt to attack this cell based on the cell-to-cell recognition performed. However, the cancer cells are continually changing the glycocalyx pattern, so it's more so like a hide and seek. So when the immune system has identified attempts to attack, it has changed already, and it's able to keep ahead of the immune system. Of course, there's more to the mechanism of cancerous cell and how they are able to keep ahead of the immune system, but this is just a very simplistic idea of how the cancer cells work. Let's take a look at the picture that we have right here just to summarize all that we have done. So in this picture here in green, so the green structures are basically the oligosaccharide chain, and we can see that they are on the extracellular side of the cell. Okay, so this would be the extracellular fluid, and then of course we can see the phospholipid bilayer. You can see the two tails, the hydrophilic head and then the hydrophobic tails, and of course we can see the transmembrane protein. So this would be the transmembrane protein transversing through the width of the cell membrane. Of course, we can see here a peripheral protein, and it also serves for attachment for the oligosaccharide chain. Also on this side, on the intracellular side, we also have a peripheral protein. Here what we can see is a channel, and it's also formed by a transmembrane protein. Alright, so far we have considered the cell, the definition of the cell theory, as well as the plasma membrane. Now let's take a look at the cytoplasm. Generally, when we're studying a cell, especially in elementary level, we'd study that the cytoplasm is this fluid substance that we have within the cell. Okay, so we define it as a semi-fluid substance that we have, kind of like jelly-like, with dissolved substances within and so on. Now that definition that we have of the cytoplasm is actually the cytosol. Okay, so a cytosol is what at elementary level would usually be defined as the cytoplasm. Now the cytoplasm is actually made of three major elements. Okay, so there are three major elements that make up the cytoplasm. This would be the cytosol, which would generally consider to be the cytoplasm. Okay, however, the cytoplasm is more than just, it's everything within the cell apart from the nucleus. So basically we have the cytosol, then we have cellular inclusions, and then lastly we would have the organelles. The cytosol is of course what we earlier defined as that semi-fluid, semi-transparent substance that we have. When something is semi-transparent, it's translucent. It has certain colloidal characteristics, and in addition to having colloidal characteristics, it also acts as a true solvent. It contains some dissolved ions, dissolved proteins, electrolytes, and so on. They're all dissolved within the cytosol, not the cytoplasm. The cytoplasm would be the umbrella term for the cytosol, the organelle, and the cellular inclusions. Now, what exactly are the cellular inclusions? Now, of course, very important for us to take note of is the fact that the cytosol is also called the cytoplasmic matrix. It just came to mind a very important concept that we need to know about this plasma membrane. Of course, we discussed the fluid mosaic theory of the plasma membrane or of the cell membrane. Now, very important are these kind of like two terms, but it's actually, okay, it's just one term, and it's the term unit membrane. Now, what exactly is a unit membrane? We'd come across this term, and it also forms one of the theories. Okay, so we have the fluid mosaic theory of the cell membrane, and we also have to some extent the unit membrane theory. The cell membrane actually behaves as a unit membrane. Now, what exactly is a unit membrane? We would come across this term mainly in histology, because of course we won't just be studying the theory of what a cell is, but we would actually be viewing tissues and cells, more so cytology. But of course, we'll be viewing tissues and cells under the microscope. And we have the light microscope and we have the electron microscope. Now, the light microscope has a lower resolution, while the electron microscope has a higher resolution. Of course, this video is not on microscopy. However, please do well to check the video on, not exactly on microscopy, the introduction to histology. We study about resolution and all of these things. Okay, so if you would like to know more about resolution, kindly check out the video on the introduction to histology. Now, the light microscope has a lower resolution. The electron microscope has a higher resolution. Now, why are we learning all of this? Why do we need to know about resolution and light microscope and all of these things? Now, we need to know this because it's important in understanding the idea of the unit membrane. Now, under the electron microscope, the cell membrane, which we also call the plasma membrane, appears to be trilaminar. Fancy term, means that the cell membrane appears to have 3 layers. Okay, so the cell membrane appears to have 3 layers under the electron microscope, and of course this would be because the electron microscope has a higher resolution. Okay, so the cell membrane appears to have 3 layers. It has 2 electron dense layers and 1 electron lucent layer. Taking a look at the orientation of the cell membrane, the hydrophilic heads form the electron dense layers, so this side and this side. Now, the hydrophobic tail forms the electron lucent layer. Okay, so basically the plasma membrane or the cell membrane appears trilaminar. However, under the light microscope, we have the plasma membrane appearing as a single layer, and this of course is because the light microscope is unable to resolve it into the 3 layers. Now, this is more of a histological concept. When a structure or a membrane appears to be trilaminar under an electron microscope and it appears as a single layer under the light microscope, such a membrane is said to be a unit membrane. Now back to the cytosol. Another element of the cytoplasm would of course be the cellular inclusions, as the name implies. So cellular inclusions are literally inclusions, and since they are inclusions, they are not essential parts of the cell. So if they are present in the cell, good and fine. If they are absent, good and fine. An example of an inclusion would be, take for example, melanin in the skin cells. Could also have certain fat droplets and so on. These are examples of inclusions. So they may or may not be present in a cell. The last element of the cytoplasm would be the organelles. So we'll take a quick look at some organelles that make up a generalized cell. So we have considered the cytosol as well as the cellular inclusions. For this video, we'll be considering just about 6 to 7 organelles in no particular order. The first organelle would be considering is the endoplasmic reticulum. We have the smooth endoplasmic reticulum, and we also have the rough ER, or the rough endoplasmic reticulum. As we'd go on to see, not only do these two organelles differ in structure, they also differ in function. The smooth endoplasmic reticulum differs from the rough endoplasmic reticulum. As we'd go on to see, they differ in structure as well as in function. The smooth endoplasmic reticulum have a smooth outlook, and the reason why they have a smooth outlook is because they are not studded by ribosomes. The rough endoplasmic reticulum, they are studded by ribosomes. The smooth endoplasmic reticulum are not studded by ribosomes. This simply means that they do not have ribosomal attachments. Another key difference between in terms of structure, of course, between the smooth endoplasmic reticulum and the rough endoplasmic reticulum would be that the smooth endoplasmic reticulum has a tubular appearance. Okay, it's kind of like formed of, very fancy word, it's formed of anastomosin tubules. This simply means a looped network of tubules. Okay, while the rough endoplasmic reticulum has, in terms of structure, it has the appearance of a flattened sac, and it's called the cystina. Now let's take a look at some of the functions of each of these organelles. Now, just as an aside, okay, organelle literally means little organs. Now back to the endoplasmic reticulum, particularly the rough endoplasmic reticulum. The rough endoplasmic reticulum, as I stated earlier, is studded by ribosomes. Of course, it's membrane bound, and it has the appearance of a flattened sac. It is continuous with the nuclear envelope. Would not be considering the nucleus. However, it's good for us to take note of the fact that the rough endoplasmic reticulum is continuous with the nuclear envelope. Now, as we'd go on to see when we take a look at ribosomes are sites of protein synthesis. And since these ribosomes have attachment to the rough endoplasmic reticulum, it makes sense that the rough endoplasmic reticulum actually performs functions of protein synthesis. So a function of the rough endoplasmic reticulum would be protein synthesis. However, it goes on in its function. It's also referred to as a membrane factory. This is so because the rough endoplasmic reticulum helps to produce, to some extent, although as we're going to see, lipid synthesis is kind of like an exclusive function of the smooth endoplasmic reticulum. However, the rough endoplasmic reticulum also helps to produce some phospholipids, particularly for the cell membrane. We'd also go on to see that although it's an exclusive function of the Golgi apparatus, we haven't studied the Golgi apparatus. However, one of the functions of the Golgi apparatus is glycosylation, which is simply a fancy word for adding of carbohydrate chain to a protein. The rough endoplasmic reticulum also plays a bit of this role. However, because it's not an exclusive function of the rough endoplasmic reticulum, that process of adding carbohydrate chain or sugar chain to protein in the rough endoplasmic reticulum, particularly in the lumen of the rough endoplasmic reticulum. It's not exactly called glycosylation. The key function of the rough endoplasmic reticulum is actually protein synthesis. Now right here in this picture we can see the rough endoplasmic reticulum. You can see how it's continuous with the nuclear envelope. So this would be the nuclear envelope, and we can see that the rough endoplasmic reticulum is continuous with the envelope. So let's take a look at the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is continuous with the rough endoplasmic reticulum, and it has a lot of functions. Well, not so many, but the very first function of the smooth endoplasmic reticulum would be considering is lipid synthesis. Of course, in terms of structure, we stated earlier that it has kind of like a looping network of tubules. It is of course not studded by ribosomes, and by implication, since it's not studded by ribosome, it doesn't play a role in protein synthesis. Another function of the smooth endoplasmic reticulum would be drug detoxification. Now, as we're going to see, there are a lot of smooth endoplasmic reticulum in the liver cells or hepatocytes. The liver is an organ in the body that plays the role of drug detoxification alongside the kidney. So it's expected that the liver cells should have a lot of smooth endoplasmic reticulum. Particularly when considering drug detoxification, we take a look at an enzyme called the cytochrome P450. Now, certain drugs that are consumed, take for instance barbiturate, is a lipid soluble drug. Now, when a substance is lipid soluble, generally not necessarily a drug, it's hard to excrete it from the body, okay, because the kidney works with water soluble substances. However, this enzyme, which is associated with the smooth endoplasmic reticulum, the cytochrome P450, helps to make such drugs as barbiturate water soluble, and by making these drugs water soluble, it actually enhances its excretion from the body. So these drugs can now be easily excreted by the kidney. Another role of the smooth endoplasmic reticulum is glycogen metabolism. Terms like glycolysis, gluconeogenesis, and so on. Glycolysis, which is the breakdown of glucose; gluconeogenesis, which has to do with the formation of glucose from non-carbohydrate sources. A very important term under glycogen metabolism would be glycogenolysis. The term is formed from glycogen as well as lysis. Lysis simply means to break down, and would go on to see lysosomes. So glycogenolysis simply has to do with the breakdown of glycogen into glucose. But of course, this breakdown is not direct. An intermediate of this process is glucose 6-phosphate. Glucose in plants is stored as starch. Glucose in animals is stored as glycogen. So of course, when glucose is needed, the glycogen would have to be converted back into glucose, and that process is glycogenolysis. Okay, and I stated that an intermediate of the process is glucose 6-phosphate. Okay, so how do we go from glucose 6-phosphate to glucose? We need an enzyme, and that enzyme is glucose 6-phosphatase. Glucose 6-phosphatase is an enzyme that is associated with the smooth endoplasmic reticulum. Hence, one of the functions of the smooth endoplasmic reticulum is glycogen metabolism. Okay, another function of the smooth endoplasmic reticulum that would be considering for this video would be calcium segregation, aka calcium storage. In skeletal cells, in skeletal muscle cells, as well as cardiac muscle cells, they have contractile functions, so contraction. Now, it's very important for us to take note of the fact that calcium ion Ca2+ plays a key role in the contractile activities of muscles. So the presence or absence of calcium determines whether or not a muscle would contract or relax, so to say. Okay, so calcium ion basically plays a role in muscle contraction, and the smooth endoplasmic reticulum helps to segregate as well as store this calcium ion. Okay, now very important for us to take note of is that because the smooth endoplasmic reticulum play a role in calcium storage, which is linked with muscle contraction, muscle cells actually have a special name for the smooth endoplasmic reticulum, and it's called the sarcoplasmic reticulum. Okay, so this would be the functions of the smooth endoplasmic reticulum that would be considering for this video. Here in this picture we can see the smooth endoplasmic reticulum, and we can see how it appears as a looping network of tubules, of course different from the rough endoplasmic reticulum. Now let's take a brief look at the mitochondria. Here we have a picture of the mitochondria. Now the mitochondria has two membranes, so it's a double membrane organelle. It has an outer membrane and an inner membrane. The outer membrane has kind of like a smooth outline, however the inner membrane has folded invaginations, and these invaginations of the inner membrane into the mitochondrial matrix. So the mitochondrial matrix would be the fluid filled path that we have here. So what we have here now, the invagination of the inner membrane into the fluid filled mitochondrial matrix is what we call the cristae, and it helps to increase the surface area for ATP production. Take for instance, we have a structure that looks like this, that looks like this, and that looks like this. Okay, and a structure that just looks like this. Notice that this structure, which has all of these curls, if we're to stretch this structure, it would be much longer than just a single length without all of these curls. So the cristae actually helps to increase the surface area for ATP production, which is the major function of the mitochondria. So the mitochondria functions ATP synthesis, and ATP is adenosine triphosphate. Okay, this is the energy currency of the biological system. We would not be considering how the ATP is produced. In subsequent videos, would be considering bioenergetics, and then would consider explicitly how ATP is produced. However, a term that summarizes the whole process would be oxidative phosphorylation. Now, in terms of why it is called oxidative phosphorylation, so basically when we have a substance like glucose, glucose is oxidized, and the oxidation of glucose would release energy. So energy is released when glucose is oxidized. Now, this released energy is actually used to phosphorylate ADP. ADP is adenosine diphosphate, converting it into ATP, which of course is the energy currency. So first we have oxidation and then we have phosphorylation, hence the term oxidative phosphorylation. Now it's important for us to take note of the fact that the mitochondria has its own DNA and RNA, and this is very fascinating because apart from the nucleus, which of course is the main structure that has the nuclear DNA and it's involved in storing the DNA of the cell, the mitochondria is kind of like the only other organelle that has DNA present in it. Now the DNA present in the mitochondria is similar to that present in bacteria, or in some particular bacteria, especially purple bacteria. Now this whole idea and the presence of DNA in mitochondria actually supports a theory, the theory of endosymbiosis. Now the idea of endosymbiosis is, it's an idea that kind of explains how eukaryote cells came about. Of course, we have cells, we have eukaryote cells or eukaryotic cells, and then we have prokaryotic cells. Now, prokaryotic cells do not have membrane-bound organelle. Eukaryotic cells have membrane-bound organelles. They have a membrane-bound nucleus. Now, the idea of endosymbiosis is that the mitochondria actually once existed as an organism. Okay, so it once existed as an organism precisely actually an aerobic bacteria, hence the reason why it has its own DNA and RNA and genes and so on. Okay, so it once existed as an aerobic bacteria and it got engulfed by the ancestors of these eukaryotic cells. Then of course it lost its other functional abilities and then it just became the mitochondria. This is the idea of endosymbiosis. Going back to the mitochondria as an organelle, between the inner membrane and the outer membrane, what we have is the intermembranous space. So this is just the general structure of the mitochondria and its function. Now, as we would go on in our study, we would get to see that the mitochondria does play a function in apoptosis. Now, apoptosis, as we would go on to study, is a programmed cell death. It's different from necrosis. Necrosis is an unprogrammed cell death. However, apoptosis is a programmed cell death, and the mitochondria does play a role in apoptosis, and the enzyme associated with the mitochondrial function in apoptosis is the cytochrome C. Okay, so we have the cytochrome C. However, for now, we have learned the overall structure of the mitochondria and its function in ATP synthesis, as well as a bit of the idea of endosymbiosis, which we said is a theory and is not a law, so it's not necessarily true. Now, going on in our study, so far we have considered the smooth endoplasmic reticulum, the rough endoplasmic reticulum, as well as the mitochondria. Let's consider some other organelles that we have in the cell. The next organelle we'll be considering is the Golgi apparatus. Okay, now the Golgi apparatus, as we'd go on to see, also has the flattened sac structure, cystina, okay, and it also has some associated vesicles, Golgi vesicles, Golgi body. The Golgi body and the Golgi vesicles form the entire Golgi apparatus. Okay, now basically the Golgi apparatus receives these proteins. Okay, so when we were considering the rough endoplasmic reticulum, we made mention of the fact that the rough endoplasmic reticulum is associated with protein synthesis because it's studded with the ribosomes. Now, these proteins that are synthesized by the rough endoplasmic reticulum, of course, we have different classes of proteins. Of the proteins synthesized, we have secretory proteins. These proteins are taken out of the cell by exocytosis. We have lysosomal proteins, and we also have some membrane proteins being produced by the rough endoplasmic reticulum. Now, when these proteins are produced by the rough endoplasmic reticulum, they are transported via transport vesicles. Okay, so transport vesicles transport these proteins from the rough endoplasmic reticulum to the Golgi apparatus for packaging. Okay, so the Golgi apparatus functions in packaging of protein or protein packaging. Now, very important for us to take note of is also the idea of glycosylation. Now, glycosylation, as I stated earlier, simply has to do with the addition of a carbohydrate chain or a sugar chain to a protein. Certain enzymes that are associated with the Golgi apparatus help to carry out this process of glycosylation. Now, of course, we have, as I stated earlier, these three categories of proteins that are produced by the rough endoplasmic reticulum: secretory proteins, lysosomal proteins, and membrane proteins. Now, these lysosomal proteins, when we go on to study about the lysosomal proteins are destructive proteins. When they are released into the cell automatically dies. So lysosomes are involved in postmortem degradation. So these lysosomal proteins, which are actually enzymes, lysosomal proteins, it's also called the lysosomal hydrolysis or acid hydrolysis. These lysosomal enzymes have to be segregated, because if they are not well segregated and they happen to be released into the cell, they would cause for the cell to disintegrate. Okay, so they have to be segregated. Now, within the Golgi apparatus, the lysosomal proteins are marked with a particular carbohydrate. Okay, so they are marked with mannose 6-phosphate in order to segregate them from the cell, and they are appropriately delivered to the lysosomal bodies or to the lysosomes. Okay, of course, secretory proteins are appropriately packaged and delivered to the cell membrane, where they get secreted outside of the cell, and of course membrane proteins as well get delivered. So the Golgi apparatus is associated with the packaging of these proteins. Now, as I stated earlier when we were discussing the rough ER, the rough endoplasmic reticulum also plays a role in the addition of this sugar group to proteins. However, the Golgi apparatus continues that process by adding some more sugar or trimming off some of the sugar chain, okay, which of course we stated as glycosylation. Okay, so what we have majorly taking place in the Golgi apparatus is packaging of the protein. There's a bit of folding that also takes place within the Golgi apparatus, so it's very important for us to take note of this. Also very important in the structure of the Golgi apparatus, we have the cis phase and the trans phase of the Golgi apparatus. The cis phase is convex and the trans phase is concave. If we have something like this, this would be the cis phase, convex, and then we have the trans phase, which is concave. Now, the cis phase is the part of the Golgi apparatus that is directed towards the rough endoplasmic reticulum. So on this side, at the cis phase, we would have the rough endoplasmic reticulum, and budding off from the trans phase, we would have all of these vesicles. We would also have the medial compartment of the Golgi apparatus. Here in this picture, we can see the Golgi apparatus. So this is the Golgi apparatus in green. Here we can see some associated vesicles. We can also see how some of the secretory proteins leave the cell by exocytosis. The next organelle we'd be considering would be the ribosomes. Now, the ribosomes are non-membrane bound, and they are made up of two subunits, a large subunit and a small subunit. These subunits fit together like an acorn. Okay, so we have something like this. This would be the large subunit and this would be the small subunit. Very interesting is the fact that the ribosome is more or less a macromolecule. This is because the ribosome is made of ribosomal RNA, but not just ribosomal RNA. The ribosomal RNA is complexed. To be complexed simply means to add. So it's complexed with proteins. Okay, so proteins are added to this ribosomal RNA. When proteins are added to this ribosomal RNA, they become ribonucleoproteins. This is basically what makes up the ribosomes, and the ribosomes are sites of protein synthesis. Now, as we would go on to see, the ribosomes could be free. These free ribosomes are just scattered all around through the cytosol. Now, free ribosomes produce proteins that stay in the cytosol, so they produce cytosolic proteins. We also have membrane bound ribosomes. It's the membrane bound ribosomes that give the rough endoplasmic reticulum its rough appearance or its studded appearance. As we would go on to see in our study of protein synthesis and the cell nucleus, the membrane bound ribosomes are actually the ribosomes that produce the secretory proteins and the proteins that would get to leave the cell. The next organelle we'll be considering would be peroxisomes. Now, peroxisome literally means peroxide body. Now, the peroxisomes have two major enzymes. They have the oxidase and the catalase. Generally, we have all of these metabolic activities being carried out in the cell, and a byproduct of some of the metabolic activities being carried out in the cell would be we have all of these free radicals. Okay, so we have the formation of free radicals. Now, free radicals have unpaired electrons. Presence of these unpaired electrons in free radicals make them to be highly reactive. It means that this free radical could go on to react with some very important molecules that we have within the cell, and they could, by implication, disrupt certain functions and activities of the cell. Now, this is where the enzyme oxidase comes to play. The oxidase, which is an enzyme associated with the peroxisome, helps to convert these free radicals into hydrogen peroxide. Hydrogen peroxide is also toxic to the cell. Now, the enzyme catalase helps to convert hydrogen peroxide into water. Okay, so it helps to get rid of the free radicals produced by some of these metabolic activities. So it's called peroxide body because it's associated with hydrogen peroxide. The next organelle we'd be considering for this video would be the lysosomes. Now, from the term lyse, it's associated with lysis, which has to do with breakdown. The lysosomes are referred to as disintegrator bodies because lysis has to do with disintegrating, and generally when we come across the term some, like peroxisomes, we said are peroxide bodies. Lysosomes are disintegrator bodies, and they help in disintegrating certain organelles. Okay, so certain organelles that are worn out. For example, we could have the mitochondria, which we earlier discussed for ATP synthesis. It could get worn out, and when such organelles get worn out, they need to be destroyed, so to say, and taken out of the cell. This is where the lysosomes come to play. As lysosomal bodies or as disintegrative bodies, they help to break down some of these worn out organelles or worn out structures, and they are able to break down a lot of different macromolecules because of the enzymes that they contain. When we were studying the Golgi apparatus as well as the Rumpf endoplasmic reticulum, we saw that we had the synthesis of lysosomal proteins, and we said these lysosomal proteins are tagged by mannose 6-phosphate and they are delivered to become lysosome. Okay, so they contain lysosomal hydrolysis or acid hydrolysis. Now, it's very important for us to take note of the fact that first of all, the lysosome is membrane bound. It has to be membrane bound in order to segregate its content from the cell. When we're studying macromolecules, of course, we have proteins, we have carbohydrates, we have lipids, and then we have nucleic acid. Now, these macromolecules, especially proteins and carbohydrates, are produced by dehydration synthesis, which is a condensation reaction. So we have a small unit, it undergoes dehydration synthesis, which is a condensation reaction. Water is removed, and then we have a big macromolecule. Now, in order for us to carry out the reverse process, we need to add water. So the polymerization is a dehydration reaction. In order for us to break it down from big to small, we would have to add water, and this addition of water that leads to breaking down. So using water to break down is hydrolysis. So we have hydro for water and lysis for breakdown. So we have the enzyme hydrolase. We have different hydrolyses that are contained within the lysosomes that help to break down different macromolecules, and it's an acid hydrolase because the environment, so the interior of the lysosome, is acidic in nature. The membrane that binds the lysosome actually contains hydrogen ion pumps. So the hydrogen ion pumps help to pump in hydrogen ion from the cytosol into the lysosome in order to maintain the acidic environment of the interior of the lysosome. It's also very important for us to take note of the fact that lysosomes play a role in postmortem degeneration. Okay, so when a cell dies, when a cell has stopped receiving oxygen, what happens is that the membrane of the lysosome becomes disintegrated, and so the enzymes, the lysosomal proteins, are released into the cell, and of course it leads to postmortem degeneration. Okay, so that's briefly on the lysosome. The next structure we'll be considering for this video would be the cytoskeleton. The cytosol is this translucent, semi-transparent substance. Okay, we saw the definition of the cytosol and its characteristics. However, it's very important for us to take note of the fact that within the cytoplasm we also have the cytoskeleton. Now the cytoskeleton acts as the muscles and the ligament as well as the bone of the cell. They help to give the cell its framework. For this video, we'll be considering three cytoskeletal elements. These elements are, firstly, we have the microfilament. The microfilament is this semi-flexible thread-like or rod-like filament that we have within the cell. The major protein that we have associated with the microfilament is actin, and we'll see this actin and microfilaments come into play in cytokinesis. In cytokinesis, which is a step in cell division, what we have is the formation of the cleavage furrow, which is this constriction midway of the cell in order to form two daughter cells. The actin filaments that form the microfilament play a role in this step of cytokinesis. The next cytoskeletal element would be considering would be the intermediate filament. Now the intermediate filament, as the name implies, they are midway between microfilament and microtubules. They also have their very important roles, and in terms of structure they are mostly made of protein fibers. That's for the intermediate filament. And lastly, we have the microtubules. The microtubules are made of the protein tubulin, and they are the thickest of all the three cytoskeletal elements. Lastly, would be considering the centrioles. Now, as we would go on to see, the centrioles play a key role in mitosis, or generally mitosis and meiosis, actually generally cell division, and they help in the formation of spindle fibers. Now these spindle fibers are actually made of microtubules, and as we considered here, the microtubules are made up of the proteins tubulin. So here, when we were considering the microfilaments, we saw that the microfilaments, their associated protein is actin. For the microtubules, the associated protein is tubulin. We also have some motor proteins as well, like dynein and kinesin. Very importantly is that the centrioles help in the formation of spindle fiber, which are formed during cell division. So far we have considered a number of organelles. Okay, going by our outline for this video, we went on to see the smooth endoplasmic reticulum, the rough endoplasmic reticulum. We went further to take a look at the mitochondria, the peroxisomes, the lysosomes, the centrioles, and we didn't actually consider the cell nucleus. A video would be made especially considering the cell nucleus exclusively. If you liked this video, please give it a thumbs up and don't forget to subscribe. Thank you for watching till the end. Bye.